Okay, so NGS will refers to a suite of high throughput technologies that allow us to sequence the DNA and RNA at a scale and a speed that was unimaginable with the older methods. So this NGS will involve the parallel sequencing of a million to the billion of DNA simultaneously, producing an large amount of data. So let's break down with an example. Like imagine trying to solve a jigsaw puzzle. So the in traditional sequencing method, like Sanger sequencing, allow you to place one puzzle piece at a time. So you have to go through the entire puzzle slowly and meticulously, piece by piece. But in contrast, NGS is like solving hundreds of the pieces at once. So, like dramatically speeding up the process and allowing you to finish the puzzle in a fraction of a time. So, like the process of NGS starts with breaking down the DNA into a smaller fragments. So, these fragments are then tagged with the adapter sequence and amplified using a polymerase chain reaction. The sequencer then reads the fragment and bioinformatics um, pieces these sequence together, creating a continuous stretch of DNA. Like think of this as using an overlapping puzzle. Overlapping puzzle pieces to reconstruct the original image. So you will get a proper. It will go and attach at a proper pace where it is supposed to be. So like another way to think NGS is like a reading a book. Like Sanger sequence is like reading one word at a time, which is slow and inefficient for a large book such as genome. NGS allows you to read the entire page at once, dramatically increasing your ability to gather the information. So NGS is also scalable with a platform like Lumina. You can choose to sequence a small targeted region like a single gene or go for a whole genome sequence project. So the versatility makes the NGS application is more diverse field and the personalized medicine to the population genomics. So in a like in a easy words, let's say there is a patient comes to a doctor and the doctor couldn't find what is the cause of a particular disorder or particular disease. So he asked the genomist to do for NGS. So what she will do, she will extract a blood or saliva from the patient, and she will isolate the DNA. She purify the DNA and go for PCR. After isolating, she'll do the fragmentation of the DNA, cutting into smaller pieces of the nucleotide, and she will go for the PCR. So while doing for the PCR, she'll add some adapter. So we will be learning about adapters and everything in the upcoming class. And once she get the whole amount of DNA, she will give it to the NGS machine. So that machine will take 1 day, solid maybe 2 to 3 days, and will give a large amount of data. So in the large amount of data about the whole DNA sequence of the particular patient, and we will just go through the sequence. So the sequence will be in the FASTA format, very raw format, and we will be cleaning that particular sequence, and we'll edit the particular sequence, and we will see the difference between the normal and the particular patient sequence. By that, if there is any mutation, if there is any missing nucleotide, or if there is any overlapping nucleotide, we can be easily identify it. So this is the purpose of the NGS, nuclear generation sequencing. Basically, it gives a faster and high uh, throughput of a our DNA sequence. So, what is the difference between Sanger's and the NGS? So, before NGS, scientists relied on a Sanger sequence. So, it is a method that is developed in the 1970s. The Sanger sequence involves the selective incorporation of the chain terminating nucleotide during the DNA replication, which resulting in the DNA fragmenting of the varying length. So these fragments are separated by size, and the sequence of the nucleotide is determined by uh, detecting the labeled fragments. So this method was groundbreaking and was used in the Human Genome Project. So, and one end of interesting fact is, so the Human Genome Project was uh, started at around 2001 or something, but no, 1979. So, okay. So when they started to get 100 percentage of the whole genome sequence, they took 32 years. But using the NGS mission, we can get the sequence by 2 to 3 days. So it's like dramatical changes, right? So that is one of the key thing why they shifted to NGS. So the NGS method was, as I said, it was very groundbreaking at the time, and it was a one that is used to Human Genome Project. So Human Genome Project took over like 32 years and cost billions of dollars, and the saga sequence is still used today for the smaller scale project, but it has like a limitation. It is slow, it is expensive, and it is impractical for the large scale genome sequencing. So like imagine trying to send a letter. The saga sequence is like mailing one letter at a time, or one letter at a time. So it can still work, but it's slow and inefficient, especially when it have to when you have a lot of letters to send. So this NGS, on other hand, it's like sending a mass mail, thousands of letters and are sent at once. So it is much faster and much efficient. So this shift is comparable to the evolution of the computing technology. So the Sanger's sequence is like using an old desktop computer from the 1990s, which is capable of running one program at a time. When it comes to NGS, it's like a modern smartphone running a multiple apps simultaneously with much greater speed and efficiency. So this is a major difference between the Sangers and the NGS, and that is why we shifted to the NGS. So next, see the history of NGS. So the development of NGS technologies mark as a major turning point in the genomic research. The first widely adopted NGS platform was introduced by 454 Life Sciences in 2005. So these many peoples are involved in development in the developing this NGS, and like which is used for the technical technique called pyrosequencing. So this technology allowed for the sequencing of millions of the DNA fragments in parallel, and the significant improvement over the Sanger sequence. So shortly after, Lumina introduced their sequence by introduced sequencing by synthesis. It is a platform which became the industry standard due to its scalability, accuracy, and high throughput. So, Lumina technology made it possible to sequence an entire human genome in just a few days, like at a fraction of a cost of an early methods. So, think of this as and the transition from the old computers to the smartphones. Look, early DNA sequence technologies were likely to bulky, slow computers, or I mean, so while in NGS, it is sleek, fast, and highly capable smartphones that can perform any task simultaneously. So today, NGS technologies have advanced even further with the platforms like specific biosciences and that is also called as PacBio, and which offers a single molecule's real-time sequencing. So that is called as SMRT Smart, uh, and Oxford Nanopore. So Oxford Nanopore. Okay, so Oxford uh, Nanopore, which provides a portable real-time sequence. So with these newer platform offers the ability to the sequence very long reads, which helps in assembling a complex genome and detecting the structural variants. So this is the history of NGS. So why we have to learn NGS? Why? What's the purpose? So NGS in NGS had a broad application across many field of science. So in medicine, for example, it is a key tool in personalized medicine. Imagine having a custom tailored suit made just for you. It's same like NGS. So NGS will allow the doctor to sequence your data, sequence your DNA and tailor the medical treatment specifically for your genetic makeup, especially in cases like cancer. So where tumors may have a unique genetic mutation. And when it comes to in environmental science, NGS is used for metagenomics, which allow us to analyze the DNA from a complex environmental samples. Like imagine trying to identify a fish species in a river. So instead of catching each fish individually, we can now take a water sample, sequence the DNA present in that, and determine all the species living in that environment, which has wide applications in the biodiversity studies, environmental monitoring, and even tracking the effect of climatic change. So NGS is also essential in the evolutionary biology, where also in forensic science, agriculture, and more. It allows us to study the genetic variation in the population by track the spread of infection disease and improve the crops by identifying the beneficial genetic traits. So how NGS will work, how the nuclear genomic sequence will, next generation sequencing is working. Okay, so yeah, next generation sequencing has completely transformed the field of genomics by allowing us to sequence DNA or RNA on an unprecedented scale. So let's divide into a step-by-step process on how the NGS will work. So NGS typically starts with the fragmentation of the DNA. So the DNA is broken into millions of the smaller pieces. So these pieces can be short as 50 base pairs or as long as several hundred base pairs. Then each of these fragments is tagged with a small DNA sequence called an adapter. So the adapter allows the DNA fragment to stick to the surface of the sequencing machine. So once the DNA is attached to the sequencer, the fragments are amplified using polymerase chain reaction. So these steps will create millions of copies of each fragment so that the sequencer has enough material to detect. So think of it like making a multiple photos of a document to ensure each pages is clear and reliable. So the sequencer then begins to read each DNA fragments through a process known as sequencing by synthesis. So during the process, the sequencer adds one nucleotide, like from ATC or G, at a time to the growing DNA strand, while the fluorescent signal are emitted. So the machine records the signal from each nucleotide, allowing the computer to determine the sequence of the base in the each fragment. Like now imagine um okay like now imagine um a large crowd of people holding a piece of giant puzzle, as each person puts the piece in the place. So the bigger picture becomes very clearer. So NGS is similar, like as each fragment in a sequenced bioinformatics software is sequenced, bioinformatics software is used to align the fragment based on overlaps, so which allowing the researchers to reconstruct the original DNA sequence. So the NGS can generate massive amount of data very quickly, with some machine capable of the sequencing billions of DNA fragments like in a single run. This capability makes the NGS suitable for everything from a small targeted gene panels to the whole genome sequencing project. So for example, let's return to the jigsaw puzzle. um, in traditional sequencing method like Sanger's, you would be putting one puzzle piece in in a place at a time, very slowly, carefully, taking a long time to complete the full picture. When an NGS, on the other hand, allow you to process thousands of the pieces simultaneously. This means you can complete the puzzle which faster, much faster, because all the all the pieces are being worked at the same time. So Like another way to think about NGS is like let's say production line in the factory. So in older sequencing method, DNA fragments are processed one at a time, much like making a product one by one on a slow manual production line. While with NGS, it's like using an automated factory that can process many product at once. So the speed and volume of the volume are drastically improved. So this is how we do the NGS. First, we'll fragment the DNA. Will this adapter tagging is the one which emits the um, uh, uh, what they call um, okay. It emits a color which they can isolate it. So to make it differentiate, we add the adapter and we'll use PCR for amplification, and we'll sequence by synthesis, and we'll assemble the data, and we'll do the speed and capability. So this is how the NGS will work. So okay, so next generation sequencing generates an immense volume of data, which can be overwhelming to analyze without the right tools. So the bioinformatics tools we use serve a specific purpose to ensure the accuracy, efficiency, and the meaningful result for the sequencing process. So let's dive into some of the most important tools that you will encounter. The first is the fast queue. So fast queue, think of fast queue as the first line of a defense in your analysis workflow. Like before diving deep into the data, fast QC access the quality of your raw data sequence reads. So it evaluates whether your reads are reliable and free of errors that might that might comprise the downstream analysis. So if we start with the poor quality data, our result will be sweet. Or so the fast CQ ensures we working with the best possible data. Another important tool is BW or the bow tie tool. So these tools will perform one of the most essential tasks in the NGS analysis, like read alignment. So they match your sequenced reads to an reference genome like in the puzzle. So without this step, you wouldn't know where your sequence fit in the context of a known genomes, and you wouldn't identify the important variations like mutation or the structural changes. So for that, we'll using the bow tie too. And we next like spat, des, and velvet. So sometimes you might be working without a reference genome. So in that case, the spat, DS, and velvet comes in a handy for a de novo genome assembly. So they will help you to piece together the genome from the scratch, like just like recognizing the puzzle without a reference picture. So this is especially useful for studying the new organisms or the environmental samples. And next important tool is the JAT-K. So after assembling and aligning your data, the JAT, the GAT-K steps in for the variant calling. It used to identify the mutation, incineration, deletion, and other differences between your sequence data and the reference genome. So like imagine you are a detective comparing a suspect statement against the truth. So this GATK highlights all the discrepancies that could be crucial to understanding the genetic variation. So why this is matter is each of these tools plays a vital role in turning a raw unfiltered data into a reliable high quality information. So if any tool is misused, or if the initial data is of poor quality, the final result will be inaccurate, potentially leading to the very bad conclusion in research or in the clinical diagnostic. So these are the some of the tools that we'll be using in the NGS analysis. So now let's see what are all the challenges in the NGS. So while okay. So while NGS technology is a game changer in genomics, it does come with a set of challenges that need careful management. So here are some of the key hurdles. So data storage or the data management. Um, Okay, the NGL produce a n number of amount of data. So, for instance, sequencing a single human genome can generate more than 100 gigabytes of data. So, managing the data requires a robust storage infrastructure and considerably computational power to process and analyze it. So, imagine trying to store hundreds of terabyte of data daily. It is a logistical challenge that must be addressed. And next, the error rates. So not all sequencing platform are created equally. Are created very equally. So different NGS technologies comes with the varying error rates. For example, Illumina is known for its high accuracy with the short reads, while Oxford Nanopore generates longer reads, but the cost of higher error rates. So understanding and compensating for these errors is very essential for ensuring the accuracy of the result. Another is the data interpretation. So even after sequencing the initial analysis, making sense of data can be very complex, particularly in metagenomics or other studies involving a mixed sample. So interpreting a result require expertise in bioinformatics to accurately um, disappear which sequence comes from which organisms. And finally, the cost of analysis. So while sequencing itself has become more affordable, the downstream cost of the data storage, analysis, and interpretation can still be very high. So the large scale projects such as population level sequencing requires a significant investment in the computational resources and specialized personnel. But how we can overcome these challenges? So we can use quality control tools. So incorporating tools like fast QC will ensure our start with a higher quality data, reducing our error propagation through later stages of analysis. And by choosing a right platform, so select a sequence platform that aligns with your specific research needs. Like for example, use Illumina for a short highly accurate reads, or Oxford Nanopore for the longer reads when necessary. And collaborative with the bioinformatics. So data interpretation is one of the most challenging aspect of NGS. So bioinformaticians can help to navigate the complexity of the data and the extract meaningful biological insight. So these are some of the challenges, and that is how we'll overcome it. So now let's see the power of real world cases. So the power of NGS become evident when we look at a real world application that have transformed the like environmental science, public health, and the healthcare. So one of the thing is the COVID-19 pandemic. So NGS was the forefront of the effort to track the spread of SARS-CoV-2 the virus that is responsible for the COVID-19. So by sequencing the viral sample from a patient around the globe, scientists were able to track the mutation and monitor the emergencies of the variant like Delta and Omicron. So the real-time genomic surveillance can crucial in guiding a public health policy such as vaccine development and the distribution. Like during the pandemic, researchers sequenced viral sample to track how the virus was evolving. This helped in updating the vaccine and informing the government on which variant poses the greater risk to the public health. So as well as in the cancer diagnostics. So in oncology, the NGS is used to sequence the tumor DNA and pinpoint the mutation that uh, may be driving cancer progression. So this approach, known as a personalized medicine, enables the doctor to select a treatment based on the genetic profile of a patient tumor, leading to the more effective targeted therapies. Like NGS allows the doctor to determine whether a patient tumor um, has a specific mutation that can be targeted by the drugs, and improving a survival rates by reducing unnecessary treatments. So in field of environmental science like in ecology, NGS is used to analyze the complex microbial communities through the metagenomics. So by sequencing the DNA from environmental sample, scientists can identify and study the diverse species that contribute to the vital processing like nutrient cycling in an ecosystem. So by using NGS to sequence a soil sample from a forest, researchers can determine which microbial species are playing the key roles in maintaining the soil health and supporting the plant growth. So these are some of the places where we are using right now the NGS. So why NGS revolutionized the genomics? So okay. So then okay. Um yes. Okay, then NGS has revolutionized the genomics in several ways, making it an indispensable tool in research, as well as in clinical diagnosis and even in the environmental monitoring. So the key difference between NGS and the traditional sequencing is its ability to handle a large scale project with the speed, accuracy, and affordability. So the NGS is incredibly faster compared to the traditional method. While the standard sequence might take weeks or even months to sequence a large genome, NGS can accomplishes the same task in a few days. So this is essential for time sensitive applications such as diagnosing a genetic disorder or tracking an outbreak of infectious diseases. And next, the cost. So when the human genome project was completed using Sanger sequencing, it cost approximately $2.7 billion to sequence the first human genome. So today, thanks to NGS, it costs around $1,000 or less a sequence less to the sequence a full human genome. So this drastic reduction in cost has made the genome sequencing accessible not only to large research institute but also for the hospitals and even the individual customers. And next is scale. So NGS can sequence billions of DNA fragments in a parallel, allowing the researchers to analyze the entire genomes, transcriptomes, or even the environmental DNA samples. Like for example, in a cancer research, instead of analyzing one gene at a time, the researchers can now look at all the cancer-related genes simultaneously, identifying a genetic mutation that may inform the treatment decision. And the versatility. So the NGS is a highly adaptive. The researcher can choose to sequence a small targeted region of a DNA or they can sequence entire genome depending on the research question. So this versatility means the NGS can be applied to everything from diagnosing a rare genetic disease to studying the microbial communities in the environment. So so for let's say imagine you are going to the tailor to get a customized suit. So the tailor makes you a exact measurement and create a suit that fits you perfectly. In a similar way, NGS allows the doctor to take your genetic measurements and design a treatment specifically for you based on your unique genetic makeup. So this is known as a personalized medicine. It has already been used, being used to design the targeted therapies for cancer patient where the specific mutation in the tumor are identified using a NGS and the treatment are tailored accordingly. So let's say you want to understand the biodiversity of the river. So without catching one observing, one or anything, with the NGS, you can collect a sample of a water and sequence the DNA present in it. So the DNA will include traces from all organisms in the river, allowing you to identify the species that is present in present in. That is what NGS allows the scientists to do in environmental research and to analyze a complex mixture of DNA from ecosystem without having the individual isolate for each species. So this is how it revolutionized the world. So let's see ethical consideration. Okay, as with any underground baking technology, next generation sequencing comes with its own ethical challenge. One of the most significant concern revolves around the privacy uh, and the taking care of the patient. So NGS will generate a vast amount of genetic data, which often include highly personal information. Imagine like having a detailed map of someone's genetic makeup, and it has relieve not only their own health risk but also the information about their family members. So this raises important question like how do we ensure that this is the data is used responsibly? Who owns the genetic information in a clinical setting? The ethical handling of a next generation sequencing data is very crucial. So if a patient genetic information were relieves a pre predisposition to a disease, should this should this inform I mean should this information be shared with the insurance companies, or should family member be noticed even if the patient chooses not to share this information with them? Or yeah, so like that. So another ethical issue involves consent. So while patient can consent a sequencing the genome for a specific study, what happens to that data after the study is completed? Can it just be a further research without their knowledge? As a researcher and a clinical working with NGS, we must always prioritize the informed consent and ensures that the individuals understand how the genetic data will be used and stored. So finally, we must consider the potential of genetic disclaimers. If someone's genetic profile reveals an increased risk, released it goes into a very risk for the certain conditions. Could this lead to a discrimination in the employment or insurance coverage? Laws like Genetic Information Nano Discrimination Act, that is GINA in the US, helps to protect against this, but it's an ongoing concern as NGS become more widespread. So the key take Okay, the key takeaway is that while NGS opens new door for science and medicine, we must navigate these ethical concerns carefully to ensure the privacy and individual rights of the protected of the person is protected. And okay, so these are the ethical consideration that we have to consider when working in a NGS. So okay, now let's um go for comparing the NGS platform. There are several next generation sequencing platform that is available today, each with its strength and weakness. So let's take a closer look at the three most widely used platform, that is Lumina, Pacific Biosciences, and Oxford Nanopore. So Illumina. So this is the most common NGS platform used this both the research and clinical setting. It relies on a sequencing by synthesis technology by offering a short reads or around 1,000 or to the 10,000 100 or the 300 base pairs. Illumina is known for its high accuracy and low error rates, making it ideal for the project that requires a precise data. So, such as whole genome sequencing or exome sequencing. So it is short read lens can be disadvantaged when dealing with the complex region of the genome. And cost wise, Lumina offers one of the most affordable options for the large scale production and large scale project. And next we have a pack bio, that is Pacific Bioscience. So the pack bio uses single molecule real time sequencing to produce much more longer reads, or often over 10,000 base pairs. This make it useful for de novo genome assembly for any detecting a large structural variations in the array. Give me a minute. Okay. Um. So the error rate can be higher than the Illumina, though the recent advancement have reduced significantly. So the PacBio is typically more expensive, but is a long read capacity, make it the go-to for the complex sequencing task. And finally, we have the Oxford Nanopore. So this platform stands out for its probability and the ability to generate the ultra long reads up to 1 million base pairs in some cases. Oxford Nanopore technology is a real time, meaning meaning that you can start and analyzing the data as soon as the sequencing begins. It is highly flexible, allowing for both a small and large project, and um, yeah, and can be used in the field due to its complete compact size. But still, like a packed bio, the error rate is higher compared to the Lumina. Though the environmental or continually being made. So the choice of platform depends on your research needs. If you require the high accuracy and the cost efficient, Lumina is the one that offer the best choice. If you need long reads for the complex genome, PacBio and Oxford Nanopore offers a powerful alternatives, and at a higher cost with the trade-off in the error rates. So Okay. So what is the future or the recent advancement in the NGS? So the field of next generation sequencing is rapidly evolving, and the future holds an exciting possibilities. So the recent advancement are pushing the boundaries of what we can achieve with NGS and the faster sequencing speeds to the more accessible technologies. So one of the most significant advancement in is the improvement in the long read sequencing technologies like those offered by the PacBio and the Oxford Nanopore. So while early NGS focuses on the short read, these are the long reads, structurally variations that would be impossible for the short reads. So in the coming years, we can expect to continued educations in the education the error rates and cost efficient, making a long read sequence more accessible for the research. So another area of innovation is the cell signaling yeah, single cell sequencing. So rather than sequencing a bulk population of the cells, these technology allow us to sequence the genome or the transcriptome of the individuals. So this is transforming field to like the cancer research and the development biology, where the ability to study the genetic variations between a cell can provide a crucial insight into a disease progression and the treatment. So it is Okay. It is also becoming more prominent, especially in a platform like Oxford Nanopore. So that is real-time sequencing. So the technology allows for the immediate analysis of the data as it is begin generated, which is particularly useful in the clinical settings where the timely results are essential. So imagine diagnosing a patient with a genetic disorder in hundreds. So instead of days, the this could soon be relay with the every time sequencing achievement. So looking ahead, the integration of the artificial intelligence and the machine learning with the NGS data sequence is a highly likely to be game changer. So the AI can help to automate the interpretation of a complex genomic data, identifying the patterns and the variations that might be missed by the human analysis. So this could revolutionize the field like a personalized medicine and um, where fast and accurate interpretation of individual genome is critical for the tailored environment. So yeah, in conclusion, the future of NGS it's incredibly promising as the technology become faster, cheaper, and more precise. We will see its we will yeah, we'll see its applications expand across the field, from space exploration to the more advanced clinical diagnostic, opening new frontiers in the physics, I mean, in the science and medicine. So so NGS has completely transformed the field of genomics by making sequencing faster, cheaper, and more scalable. Whether you're working on a whole genome sequencing, transcriptomics, or the environmental studies, NGS offers a powerful and the flexible tools for your research compared to the traditional method like Sanger sequencing or the Sanger sequencing. So the NGS is far more efficient and affordable, making it accessible to the wide range of researchers and the clinicians. So in our upcoming session, we'll dive deeper into the technical aspects of the NGS, including a sequence alignment, de novo assembly, and the transcriptomics. So you'll get a hands-on experience with the bioinformatics tools like to analyze the NGS data, and by the end of this course, you will have a skills to apply on the NGS in your work. So basically, this is NGS.